Hey guys, welcome back. Today we're going to be looking at making recombinant DNA molecules. So there's a lot going on in this chapter, but specifically we're going to be focusing today on targeting our gene of interest, the types of vectors or these constructs that we're going to make. We're going to be dealing with restriction enzymes and how can we use this combination of these vectors and restriction enzymes to get our insert into the vector correctly. So there are a couple of types of vectors that we're going to be talking about. So when we talk about a vector, what we really mean a lot of the times is going to be something like a plasmid. Okay, like we saw in the last video, that one we were using these plasmids to make knockouts in yeast and mice. Okay, so a lot of the times what you can envision with a vector, it's going to be something like a plasmid. It's going to be used to sort of hold the gene of interest for a couple of reasons. Okay, we can have shuttle vectors, which are going to be used to literally shuttle back and forth between different kind of hosts. So maybe we have to start in bacteria, then we'll drop it into a plant, or that agouti mouse from last time. Shuttle vectors have to be able to help us get not only from the organism we start with, but maybe a different target organism later on. So we'll talk about those at the end a little bit more. What we're really going to look at today are these expression and transcribable vectors. So there's a lot of overlap between these types of vectors, but the idea is that they have to be able to create messenger RNA that can be used for maybe protein expression, but other types of things that are RNA-based as well. We have to be able to get the cell that it's in to transcribe it. So that's what we mean by expression vectors. So we're going to be dealing with that. I think first is one of the most straightforward or quintessential kind of examples of making these recombinant DNA molecules. So let's take a look at some of these expression vectors. So as I mentioned, when you think about these vectors, you can envision, again, sort of a circular piece of DNA. A lot of times these, again, are based off of bacterial plasmids. So that doesn't necessarily have to be the case, but this is kind of what you can envision. And so it's going to have several characteristics, some of which are going to have nothing to do with our target gene, okay, but they just have to be there so that we can do business in the bacterial cell or wherever we're going to put this. Some of it is going to have to do with getting our gene of interest into this vector, okay? So let's deal with a couple of these things. So when we think about the actual expression part of this, okay, so what we're going to have, going from left to right, we're going to have a promoter. So I really hope you guys remember what that is for. That's going to be where our transcriptional machinery is going to assemble. So these will be those transcription factors that we need. This is going to be where our RNA polymerase is going to land, okay? So next in line, we have the Shine-Dalgarno sequence. This is not something that we really need right now for transcription, but it's going to be part of the RNA so that we can get a good ribosomal binding site. So that's what that's about, okay? And then let's just do the terminator. We'll come back to this middle bit. That's going to help terminate transcription. So that will create sort of a transcribable unit between the promoter and the terminator, okay? So what's between those two will ultimately be part of our RNA. Okay, so what's with this multiple cloning site? Okay, our multiple cloning site is going to contain several different types of restriction enzyme sites. So I hope that you guys remember about restriction enzymes from some of the earlier assignments that you had in class. Okay, we're going to review them a little bit today and kind of zoom in on what they're doing. But you should know that these restriction enzymes like to cut open DNA whenever they encounter a sequence that is their preferred binding and cutting sequence. So each one of these enzymes has a different place that it likes to land and cut. Okay, so we've got here 3 examples. KPN1, that's a pretty common one, BAMH1, SAL1. These are all going to be pretty common restriction enzymes, and these sites are very close together. So this multiple cloning site gives you some flexibility. So we'll be seeing in the next couple of slides how to prepare our actual gene of interest that we want to express in this vector to be dropped down into this multiple cloning site. Okay, but having different restriction sites allows us some flexibility so we can choose whichever of these enzymes is going to best suit our needs. Okay, so we'll be talking about that more as we go. Some other things, some attributes that you might have noticed are things like the ORI sequence right here. This is the origin of replication. This is here because imagine bacteria divide. This has to be here to remind them that they need to make a copy of this too as they divide so that you don't dilute out however much plasmid you put in between the bacteria as they start to divide. We'll also have a selectable marker, just like we talked about in the last video. So this particular one is for ampicillin resistance. It's an antibiotic resistance. This is a pretty common one. Okay, so there's just kind of a lot going on here, just some stuff that's day-to-day business for a plasmid. Okay, and then the stuff up here that's going to have to do with actually getting expression from this vector. So let's step away from that for a second and take a look at our gene of interest. Okay, so what kind of genes would you imagine would be a gene of interest? Okay, this can be anything, again, that you want it to be. So if you want to see what happens if you produce a lot of a protein, then you might put that in. If you want to harvest protein, so for example, we talked about using bacteria, using E. coli to produce things like insulin. So insulin's protein-based. Maybe we drop the gene down in there for insulin so that we can get these bacteria to create a large amount of this protein for us. So depending on what you're trying to do, there's a lot of reasons you would pick a gene of interest. Okay, so if you want to imagine this is something like insulin, because we've talked about it before, then that's a fine example. Okay, so some attributes have to be present. Okay, so in this case, restriction sites on either side of the gene. So our gene of interest here actually being just this darker orange part. These lighter parts are going to be sort of the rest of the chromosomal sequence, or the PCR that we amplified up. Okay, so this is like extra that we're not going to need. Another thing to note is that restriction sites are very common. Okay, depending on which enzyme specifically you're looking at, there are restriction sites for all types of enzymes just like lurking all along the sequence. Okay, so we know we're going to need BAMH1 sites here. Okay, but there can be all kinds of restriction sites. One that we're going to be using a little bit later at 2. Okay, so we don't need to worry about it right now, but I do want to point out that it is more towards the 3' end of this gene of interest, 5' end or the beginning of it over here. This is the end, and we'll see that this is going to be very close to the end of this sequence. Okay, so for right now, we're not going to worry about that too. We'll come back to that in a minute. Let's now deal with our BAMH1 sites. So let's drop some actual enzyme into this. So here is some BAMH1 enzyme glomming on to the sequence that they prefer. So that's marked again there as BAMH1. We have to add the actual enzyme. They cut there, and we've trimmed off that sequence. So I want to zoom in on the ends of this gene of interest. It looks really blunt here, like we've just cut it, but that's not exactly what's happened. So if we take a look at the close-up, again, where this is going to be the end, the 5' end, this will be the 3' end of that gene of interest, just as we've trimmed it. The restriction site actually has cut it so that it's uneven, okay? So it will cut kind of in a Tetris shape, okay? So we cut, 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 right? And these are called sticky ends, and we'll see in a minute why that's important, okay? So let's go back now to our vector, that expression vector that we previously looked at, okay? So we've more or less prepared our gene of interest. We have to do some stuff to this vector as well. Okay, so here is our vectors. So again, this is kind of an abbreviated version of what we explored on the first couple of slides. We've got our promoter, our terminator, presumably some type of selectable marker, and origin of replication or ORI sequence somewhere in here that's not marked. And you may notice we've marked an @2 site. Okay, we're going to use that, but not yet. Okay, the multiple cloning site will contain, again, multiple things like KPN1, SAL1, all those things. BMH1 we're going to be using right now. Okay, so notice that's the same enzyme that we use to trim up our gene of interest. Okay, so I've kind of created a more general version of that. It's the exact same thing, except a little more general. Tried to kind of color this pink where the multiple cloning site will be. Here's that BMH1 site. Okay, so what we're going to need to do next is actually open up this vector. So in order to drop DNA down into it, we've got to get this open. So we're going to use our BMH1 just like before. If we zoom in on what's happening, you'll notice maybe you don't have that sequence memorized, but that's okay. We're going to cut, 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 just like this. Okay, the enzyme is going to recognize that sequence. And what we end up doing is just opening that up. Okay, and what that's going to look like on our vector is we've opened it. One thing I want to note, when we started out, this vector was 3,500 base pairs. Okay, so that's how big this was, 3,500 base pairs. It is still that length. We have not removed chunks from it. We've simply sliced it open, and it's allowed to just come open. Okay, so we've opened that up. We had previously prepared our gene of interest, so we can literally now drop that in. Okay, and so what that's going to look like, we actually zoom in again on the sequence. Remember, we prepared that gene of interest using BMH1 as well. So it's also got sticky ends, which would be complementary to the ends that we've opened in our vector. So here's our vector end. Here is our sticky end from our gene of interest. And notice G, A, T. Okay, see, we've paired these up. All right, same thing here, paired this up. Okay, that's why this is so important, so that we can actually create this sort of base pairing situation where it will want to at least use hydrogen bonding to adhere to the sequence. So that's what's going on right at these junctions. This is still not a continuous piece of DNA until we add ligase. So yes, this is the same ligase, sealing it up just like in DNA replication. Okay, and so that's great. In principle, we have finished adding this gene of interest. And again, you can see I've used as a landmark that @2 site that was originally present in that gene of interest. There, of course, was another one over here in our plasmid, but I've not marked that one. Okay, so something important to think about is the actual nature of these sites. Okay, so once we've added ligase, they're sealed in. But notice these sticky ends are like G, A, T, C. If we come this other way, G, A, T, C, it's the same. So what that means, let's zoom in again. This is the original way that we put it in. There's actually nothing preventing this from flipping around, okay? And so you can see I've just actually rotated this. So this is kind of upside down the way we had it before, but the beginning of the gene is over here now, okay? And it can still base pair just as easily. Our eyes care if this is upside down, but these hydrogen bonds don't actually care about that, okay? Because they'll still be in contact with each other. So there's more or less a 50-50 shot of this, okay? We saw last time how much work it was to get these types of experiments going for like knockouts in mice. You really want to set yourself up for success, okay? So we want to do some checks to make sure that we put this in the correct way, okay? You can see that landmark, that @2 site that I've used as a landmark, it's now over here. This is backwards, okay? And this is a problem because our promoter is going to be here. We'll start transcription, and we are transcribing this backwards. So that is not going to make any kind of protein that we even know what it's going to be. Okay, it's not going to be functional. It's not going to be using the codons. The RNA polymerase doesn't know it should have started at this, which would have been the 5' end. Okay, so we're going to use this @2 site on the next couple of slides as a diagnostic to make sure we put this in the right way. Okay, so let's take a look. All right, and yeah, we're going to use a gel. So what we'll be doing is taking a subset of our plasmid, and now I've actually added numbers to map this out, okay? So you can see, I told you guys that this vector was going to be 3,500 base pairs, okay? So from BAMH1 site to BAMH1 site where we cut it open, 3,500 base pairs, okay? We knew from looking at our gene, okay, and I mean, I've put this on a previous slide, but because we know what gene we're trying to put in, we know the length of this sequence, okay, and this is going to be 1,800 base pairs at 2 site, and then another 200 base pairs, so a total of 2,000 base pairs. So in total, we're going to have our 3,500 and our 2,000 base pairs here, okay. So what we're going to do, we're not going to use BAM1 anymore, okay? So we're going to ignore that, okay? So they're still there, it's just a sequence, but we don't want that because if we use that enzyme, all we'll do is cut this back open, and that's not helpful. We're going to use this @2 site we have here and this one that's here in this vector to help us understand how we've positioned this, okay? So if right now, this is the correct orientation. Okay, this is the way it should have gone in, what we hope for. Let's add some @2 enzyme. It will cut at the designated sequences, and it will drop these 2 guys somewhere else, okay? So let's do a little math because you know there's a gel, so we want to know what the length is going to be. All right, so this piece here, the top, to be 500 plus 1,800 gives us a length of 2,300 base pairs. At the bottom, we're going to have 200, the length from here to here, plus the rest of this 3,000. That gives us 3,200 base pairs, okay? So now's a good time to pause that video and think about how this will look on a gel. Okay, so let's take a look. All right, and you can see that that will come out. We've got 1 fragment at 2,300 somewhere in here, okay, and 1 fragment at 3,200 somewhere in here. Okay, so what we need to do in order to really understand this is compare this to the incorrectly positioned vector, okay? So let's take a look. Notice, again, our vector is the same length. From BAMH1 site to BAMH1 site, 3,500, okay? Here's that @2 site here, okay? But now notice this gene of interest is backwards. This @2 site, which should have been on its 3' end, is now over here, close to the promoter, okay? So let's go ahead and get rid of those BAMH1 sites because, again, we don't care about them right now. If we use them, we'll just release this back out of the vector. It's not diagnostic. Okay, so let's use the @2 sites. Go ahead, open that up, and you can hopefully already see a pretty large difference here in how this is going to work out. Okay, so this piece here was 200 base pairs. This piece here was 500. Okay, so our 500 plus 200 is 700. Okay, and down here, we've got all of this 1,800 stretch plus this 3,000, okay? So that leaves us with 4,800 base pairs. So one more time, pause that video. How do you think this will look on this gel? Okay, let's take a look. Again, we had 1 fragment that was 700 and 1 fragment that was 4,800. This is what we're going to see, okay, and you can see a pretty obvious difference. All right, so we would do this as kind of a step to help us know, again, that 50-50 shot, which one we actually got, what orientation we actually got. Again, we would sequence these too, okay? But sequencing is a fair amount of work relative to a restriction digest on a plasmid. So we only want to sequence the ones that we know are in the correct orientation to begin with, okay? So we know it wouldn't be these guys here. All right, so once we do have the correct orientation, all right, we're ready to start making protein. So here is our gene of interest in our plasmid, in our vector, okay? And again, if we want to make something like insulin, we can just drop this down into a bacterial cell, okay? So we can start creating protein. We can isolate this out. We can use this to transcribe things. This is an expression vector, meaning we're getting some product for it, okay? So sometimes if we're dealing with things like shuttle vectors, sometimes what we want is just to use the bacteria as sort of an intermediate to make more plasmid, okay? And thanks to that ORI sequence, whenever they divide, they will keep making copies of that plasmid. They'll divide again and again and again and again until we get a lot of copies of this plasmid. And if we don't care about expressing anything from them, if we ultimately are using this as a shuttle to go to a plant cell, something like that, then we will isolate the plasmid out. So we'll see that on the next slide. Okay, so here's all our plasmid. In principle, this would be in a tube, okay? We would undergo transformation. So this would be similar to the GMOs that we talked about previously in the semester, okay? So we would take our plasmid, it would, however we've designed this experiment, be dropped down into a plant cell, okay? And then hopefully, if we've done this correctly, we'll end up with this gene of interest being incorporated into our actual chromosome, okay? Another example of this would be, again, putting this type of construct, for example, if we're going to make a knockout, we can put this type of construct into those mouse embryonic stem cells. So here's that agouti mouse from last time, okay? And so this can really be related to things like CRISPR as well, because in order to get that CRISPR machinery, that Cas9 protein, that guide RNA in position, we're going to have to drop some type of vector, some type of construct into the cells. So that's how this has to work, okay? So it doesn't have to go into a bacterial cell necessarily, but we need to consider what the target is, how exactly we want it to reproduce, how we want to make more copies of this, if we want it to express a protein. Okay, we have to very carefully plan the way that our construct is designed. Okay, so some conclusions from today. All right, now we know how to really prepare these recombinant DNA molecules. Okay, so we know what different vectors are for. We've talked about expression vectors and shuttle vectors, where the intent is kind of where you want something to be expressed or knocked out. Okay, so if you want something to make proteins in a plant cell or a mouse cell, you don't have to engineer the promoter so that a bacterial cell will recognize it. You just have to make sure they're going to replicate it so that you're able to eventually transfer it. Okay, if you care about making a lot of recombinant protein in bacterial cells, then you definitely want to really drive up that expression for bacteria. So you just have to think about the attributes that you need. We've also learned to use restriction enzymes not only to prepare these vectors, but to check them for accuracy. Okay, and we can use recombinant molecules in all types of assays. So we've really been secretly talking about these kind of all semester. So making those constructs in the last video for homologous recombination. If we want to make just RNA molecules like guide RNAs for CRISPR, if we want to make modifications in plants or animals, we have to know how to deal with these recombinant DNA molecules. So next time, we will be learning a little bit more about using these expression vectors to find genes that we've been searching for, okay? So this will be a little bit different than the genetic mapping type of thing, but we'll be looking for particular genes in the genome. Okay, so I'll go ahead and in here with our biology joke of the day.